Okay, question 21 of homework 4. So let's see, when you examine the DNA structure of the locus that contains the DRD5, you see something unexpected. So perhaps you don't know this answer because you didn't know what is expected. Let me draw what would be something that I would consider that's a normal. So this is one DNA strand. This is the other DNA strand. So when we did gene prediction, we'd say like, oh, if this sequence here has some mRNA molecule in that organism, then this DNA sequence here is the coding DNA for gene, let's say gene 1. And then gene 2 could be downstream from it, like on the same strand. That's gene 2. And then maybe gene 3 is on the other strand, gene 3. But normally for eukaryotes, there is space in between them. For prokaryotes, that intergenic space can be very small, and then sometimes they can even overlap by a little bit. But for eukaryotes, normally you see space. Now, what is so shocking about that gene is the fact that, let's say that I'm going to draw a bigger box in another color, so you know that's the one we're talking about. All right, so let's say that this is the DRD5 gene that you're studying, DRD5. And then it showed that there is another gene in, I don't know, let's say in here, on the other strand, which is actually much bigger than this. It was that SL whatever. And that gene that has some part of the introns of this, or this DRD5 is encoded within the intron of this sequence, but of course not on the same strand. So that's the part that is surprising. And the way you would see that is, let me show you the screen again so that you see like how do you figure that out. There are different ways you can see it. The easiest way is they overlap in here. And then the other way is when you look at the DRD5, there is also this other gene, SLL, whatever. And you're like, how can they be on the same spot, the same locus of the genome? That's weird. But one is pointing right, the other one's pointing left, which means one is encoded in one strand, the other one is encoded on the other strand. And the DRD5 is encoded within the intron of this gene here. So if I expand this gene, then I get all these lines here, which are pointing left. That's how I know they belong to this gene. And the skinny lines are introns. So the DRD5 doesn't have introns, but it is encoded within the intron of this other gene. And that is what is unexpected about that.